questions and answers at the end. So I am really, really happy that Matthew accepted my invitation, and I can hardly wait to find the story behind the demon fish. So Matthew, the floor is yours.
Thank you, Emma. Yeah, greatly appreciate the introduction, the invitation to chat with you all about our discovery here. I'm gonna try to share my screen. Let me see if I can get this to work. Let's see, window. Can you all see that?
Yes.
Hopefully. All right. I think that's working. I can get started. So yeah, before I get really kind of get into the discovery itself and kind of how this kind of transpires, I just want to recognize all the co-authors on our recent paper. So I run the Cave Biolab at the University of Alabama in Huntsville. This is in Alabama, in the southeastern United States. But several other folks were involved, including John Armbruster and Pam Hart, who were involved a lot with the morphological work in this study. Michael Sandell and I, we kind of led efforts with respect to some of the genetics and my genomics, and my lab, in particular, my graduate students, Jacob, Eric, and Lael in particular, were involved in some of the expeditions, and then Coral and others were involved with some of the lab work. And then I also want to recognize Dr. Matt Kendeas, who was our contact at Redstone Arsenal, which is a U.S. Army military installation where the discovery was actually made at. So he was also involved in the study. So a little bit of background for those of you who probably aren't intimately familiar with where caves and karst are in the United States. So this is kind of a karst map. The blue there represents some of the carbonate karst in general. And you probably can't, I don't know if you can see my cursor or not. I'm going to kind of circle it here and I'll kind of highlight it. The area that I live at is kind of right there. So it's the junction of kind of Tennessee, the states of Tennessee, Alabama, and Georgia. And it's kind of where two karst regions come into contact, the Appalachian Valley and Ridge and the Interior Low Plateau region. And in my area, so Alabama has about 5,000 caves. Just north of Tennessee, they have 12,000 caves. And so it's a really karst-rich area, a lot of cave systems. And with that, from the kind of biological side of things, this is where my expertise is and my interests largely are. There's been, a fairly long history of biological cave exploration, right, in this region. Where I live in particular is the city of Huntsville. So Huntsville is the headquarters for the National Speleological Society here in the United States, and it's been there for several decades now. And this is just some of the creatures that we have kind of in our area, and many of these are kind of analogs to species that you can find, in the Nerkarst in Europe, for example. I mean, we have pseudoscorpions and spiders, right, and beetles, and varieties, and snails. There's some of the terrestrial species here, harvestmen, millipedes, and then some of the aquatic species. Some of the things that you don't necessarily have, things like cavefishes. Although talking to Emma, maybe you will. We'll find out. Looking forward to hearing about that. But we have cave crayfishes, flatworms, aquatic snails, and lots of different types of crustaceans like isopods and amphipods, ostracods and whatnot. We have a few salamanders, a little more cave salamander diversity here in the U.S. compared to Europe. And here's these kind of major karst geographic regions that past authors, if like Dave Culver and folks, have identified in the past. And so we're kind of where I'm at right now, in northern Alabama, where I'm at that southern edge of that blue area, that interior low plateau. It's kind of the geologic setting of the discovery. Just a little bit of quick review. This is a core diagram just to kind of show you each of those karst regions is identified there at the top of the circle here. And then some of these taxonomic groups, just kind of highlighting some of the diversity that we have here in the U.S. So my lab kind of led a paper published last year kind of summarizing our cave obligate biodiversity. And so in terms of terrestrial species, we have a good number of beetles. A lot of those are in the eastern United States. Roughly about 200 or so of those, and then some other groups are fairly well represented. We still have a lot of unknown biodiversity as well in terms of aquatic species there on the right side. So most of our diversity is amphipods. I think much like Europe, we have a lot of isopods as well. This is kind of showing a heat map here, the eastern U.S., some of those major karst regions. And so the warmer colors represent areas that have greater species richness in terms of cave obligate species, right? These are the species that are adapted for living in subterranean habitats like caves and wells and whatnot. And you're not usually going to find them on the surface, or if they are found there, right, they're not going to be there very long, right? They're adapted for living underground. They're often blind, often depigmented, right? Have other adaptations for living in complete darkness. And so where I'm at there, that kind of bright red cluster is again kind of an area of high species richness, and this is for terrestrial species, also for aquatic species. It's a little more nuanced there, spread out there. Southern Indiana, Kentucky, the Mammoth Cave region. I'm sure you've probably heard of Mammoth Cave. Definitely a hot spot of biodiversity here in the United States. And this is kind of showing where there are endemic species, so species that might be found from a single cave system, right? A really small geographic area. And an area that, again, that kind of highlights, that stands out, right, is the same area in northern Alabama, kind of where I live. All right, so kind of moving forward, another common theme that we see in cave biology, in general, is this discovery of what we call cryptic diversity. This idea that you have species that look really, really similar to each other. In some cases, can be even identical morphologically, right? The characters that we typically use to describe species. Turns out that genetically, right, these can be really, really genetically distinct. In some cases, may have been isolated for millions of years, yet look almost identical, right? So we call this kind of this cryptic diversity kind of phenomena. So my lab works with a lot of different groups of organisms. So we see this phenomena in cave crayfishes. One example is a species that's found down in Florida, states of Florida and Georgia, Cambarus cryptodites. We're finding evidence of significant cryptic diversity in this group. Our little millipedes. So this is we have a genus called Scoterpes. They're about a centimeter in length. They're small little hairy guys, kind of fragile. They all look really, really similar to each other. There might be, we're estimating anywhere from like 15, possibly 30 different species that might exist, and several of these are undescribed. And what I'm going to talk about today, this new genus of cavefish that we discovered. In this case, kind of a different type of cryptic diversity in the sense that we weren't necessarily, when I say we, I mean myself, other scientists, other cavers who had access to the cave, just weren't really paying, I guess, enough attention. So the group of cave fishes that I want to introduce to you are in the family Amblyopsidae. They consist of a surface species, which is probably also a species complex. This is called the swamp fish. They're nocturnal in nature, have small eyes. They like to spend their day underneath leaf litter and whatnot in slow-moving streams. Not associated with caves, or springs at all. You have the spring cavefish. So this is a species that can be found on the surface in springs and spring runs, but also can occasionally be found underground. And then you have a variety of different cavefish species. So some of the genus Amblyopsis, there's two species there, the northern cavefish and the Hoosier cavefish. You have the Ozark cavefish. This is a United States federally endangered species. It's probably also species complex. Southern cavefish in the genus Typhlichthys, and there may be as many as 12 or 13 probably species within this group, and about 10 of those probably remain to be described officially. And then before our study here we're talking about today, the Alabama cavefish was only known from a single cave system in northern Alabama. Sometimes people give it the common name the duck-billed cavefish because it has this really pronounced, elongated head, flattened head, and giving it kind of a duck billed appearance. So kind of fast forwarding a little bit here to the cave system that we're going to talk about. So it's a small little cave that's located, really in the heart of the metropolitan area of Huntsville, city of Huntsville. So Huntsville is not necessarily the biggest city in the U.S., but it is the biggest city by population in Alabama. What's a major feature, though, is a military base known as Redstone Arsenal. So, if you're familiar with the history of the space race between Russia and the United States and the Saturn rocket that was developed, that ultimately, took astronauts to the moon, it was developed here at Redstone Arsenal. So that's kind of the history. So that military base by Warner von Braun, he selected this site, was set up in the 1960s, and during this period. And so cavers didn't first learn about the cave on the installation until about 1973. So in terms of, some cave systems, not the most well known, but still we're talking, almost, 50 plus years of knowledge. So it is restricted. It's in a restricted part of the base that was formerly used for testing ordnances and the development of things like DDT, other maybe chemical munitions and whatnot. The cave's developed in the Tuscumbia limestone. It's Ordovician aged. The cave system itself is not very big. It's only about 450 meters of map passage, and most of that, and kind of what we spend most of our time in terms of looking for some of the aquatic species, is a room that we call the shrimp room. It's about 60 meters long or so, but the cave system itself is all really, really low. It's a sinkhole in the woods. You drop down into a passage that is usually water filled, but it can fluctuate depending on the time of the year, if it's dry versus wet season. And there's only a few areas that I'm about 6 feet tall, 1 point some meters or so, and I can only stand up straight in a few places. So if you're shorter, maybe you can stand up in a few more areas, but a lot of it's stoop, it's crawl, it's wet, it's muddy. For most people, not the most enjoyable, cave system to visit. It does get flashy in terms of rain. It's a really shallow cave system, so it can fluctuate depending on the amount of rainfall by almost 4 meters in terms of water levels in the cave system. And the reason that it's kind of on our radar as a biospeleologist is because it is home to the largest population that we're aware of another federally endangered species, the Alabama cave shrimp, which is just found in a few cave systems around the Huntsville area. And again, that was initially discovered when they first started to kind of map and explore the cave back in the early 1970s. Here's what the entrance looks like. It's not a majestic entrance and a bluff or a real scenic. It really is just a little hole in a small little knoll in the woods on the western part of the installation there, the buffer zone, in general. But basically, you drop down that little slot entrance and you kind of go through your little chimney area. That's gets you down to the water in the main passage, right away. Here's the map. So the entrance is there, kind of here at the top, small little entrance, and then you're really kind of in it. And then there's that big shrimp room. It's fairly wide, fairly long, but it's low. You can see these are in feet, 4 foot, 3 feet. The whole western, essentially the left part of the map here, there's a little tight crawl that you can go through. This is all basically above the water table. It's all really, really dry. We typically don't, when we do our cave surveys here, we don't really focus on that area. We kind of drop the entrance, and this is how most biologists have studied the caves, other scientists, and have studied the shrimp room, that area, in general. So, a little bit more about the biology of the cave system. As I mentioned, it's because of the presence of the Alabama cavefish and being a federally listed species. There's been some long-term monitoring efforts by biologists starting about 1990. Early on, this is studies that were conducted by the Geological Survey of Alabama biologists. We're going about monthly, assuming water levels would allow it. And then sometime in I think around 2010-ish or so, I think they transitioned more to quarterly surveys, so 4 times a year. But regardless, right, lots and lots of historic trips to count shrimp, specifically counting the aquatic fauna in general. My lab started doing work there in about 2019. We've done about 9 different, biosurveys, right, biological inventories focusing on, again, the aquatic fauna. We had known about another species of cavefish there, the southern cavefish. There had been previous collections, all the museum collections, and we checked this in the study, were all this species. We never found this other species that I'm going to talk about, introduce you to here in just a moment. So for all intents and purposes, right, there was not any sort of expectation that, right, we would find a new cavefish species in this cave system. Having more than one cavefish species, because cavefishes typically are a top predator in aquatic cave ecosystems, is really, really rare. You really kind of have to go to Southeast Asia, really the South China karst. We have one cave system here in Alabama with the southern cavefish, Alabama cavefish could occur, but it's really, really rare. Other instances, there's just not much, knowledge of where you have these species maybe co-occurring with each other, living in the same exact pools, right, and interacting with each other. So fast forward a little bit, some of the other cool species that we find there. We have, pseudoscorpions is an undescribed species that hopefully we can get someone to help us work on and describe here, from the cave system. This is an undescribed terrestrial isopod, essentially, a cave roly-poly. We don't have quite the diversity of terrestrial isopods as you all do in Europe. This was a silverfish, so this is technically an insect, sometimes called bristles. This was a species that we actually described a couple of years back. The type locality is also bobcat cave, so something new to science, and actually a new genus of this invertebrate, Spinalicta albumensis. This is its range. So in green is the new genus, and it has relatives right in the Ozarks here in blue, and then get in Texas and out west. So really kind of an odd, odd distribution in record, and likely because again, this is a group that probably is more widespread, and we just have not ever really paid attention to it. It's a small little critter. It's has relatives that are leaf litter inhabitants on the surface. But gone kind of then in February 2025, we were visiting the cave to download some data from some data loggers and then conduct an aquatic survey like we usually do. So we're counting the cavefish that we see. We're not catching them at the time. We're counting cave crayfish that we see, shrimp, and other life. Well, I was in the back of the cave, kind of at the end of the shrimp pool, and the water was up fairly a good amount. But I had my underwater camera and I was trying to take a photograph of this really large cave crayfish. And in the process of kind of bending over, not trying to get in the pool. The reason we don't usually get in the pools too much is we don't want to disturb the shrimp. They're kind of fragile. I slipped and I fell in, and I'm a big guy. I'm going to use American terms. I'm 6'2", about 250 pounds. And so I made a big splash, kicked up a lot of silt. Underneath this ledge, this fish that you see photographed right there popped out. And it's like, oh, that's a cool fish. I thought it was a southern cavefish. Wasn't paying attention. Just took a couple photographs. I took a little video and didn't think anything of it. I didn't even look at my photographs until like 5 or 6 days later. And it was not until then where I pulled them up on my computer and started looking to realize I was like, hey, the head shape looks a little different. Oh, the fin rays don't look the same as what a southern cavefish should have. So this fish that's photographed, now we recognize as the demon cavefish, has unbranched fin rays, whereas southern cavefish and most other amblyopsids have branched fin rays. So they start out kind of like a tree, right? It's like a tree, and they start to spread out as they get to the margins of the fins. And there's some other little differences. And so it was at that point, I was like, I sent some of the photos to some colleagues, John Armbruster at Auburn University, Pam Hart at Alabama, and it's like, hey, does this fish look any different to you at all? And they kind of noticed like, oh, it does look a little bit different, but we weren't thinking anything at the time of like, oh, this is a new genus or something really like that unusual. I thought, oh, maybe it's a weird variant or whatnot. They recommended we try to go back and actually collect some specimens, and we were going to send them to John, and he's going to do some morphological work, some micro CT scans. Well, the weather forecast wasn't looking very good. We had a big rain system that was coming in, and if we get a big rain there, that cave can probably be sunk for a few months. So we scrambled, and with our contact there at the Arsenal, Matt Candeas, we were able to get in just a couple days later, and that's when we really started to pay attention to the fish and realized that, oh, this wasn't just some one-off rare fish in the cave. I mean, it's rare in the sense it's really known from this cave system, as far as we're aware right now. But we saw, I think, roughly about 20 fish in total, and like half of them were this new thing, what looked to be this new thing. And we were just really kind of floored by that. It was really surprising. And so I wish I could tell you the story that this is a well planned out expedition. We're going to the furthest reaches of a multi-kilometer cave system that took days to explore and finally found this thing. But it was honestly serendipity. It's because I was clumsy and I fell into a pool that we were able to make this discovery in general. So we collected a couple specimens on that second February trip. Did some counts, and that's kind of got things started on trying to better understand if this was a new species or not. So I'm going to show you a little bit of footage. This is the footage actually I took on that first trip, again, that I didn't pay attention to. This is the demon cavefish. This is kind of what they do. They just slowly move around, the bottom or on the rock wall, flip their pectoral fins, and have a slow kind of existence. But you can see it kind of has a bit of a hump on its back. Behind the head there, its head's a little bit flattened, but not to the same degree as that Alabama cavefish that I mentioned that kind of has that real duck-billed appearance. So that was that first video, yeah, that I took there. And so, through some additional subsequent work, right? This is a stygobiotic fish, lacks eyes, completely. Actually has significant changes to the cranial morphology, the bones of the head, in general because of that. Coloration wise, it unlike the southern cavefish, and I'll show you a couple pictures here in just a moment, it lacks any sort of melanophores. So a lot of cavefishes actually technically aren't albinos. They might just have fewer pigment cells, or they might be smaller pigment cells. This species appears to be an albino. Doesn't appear to have any of that pigmentation. That reddish area that you see there, right, you're actually like seeing through the skin. It's like semi-translucent. But in general, even in your cave-like lights or dive lights, it kind of has this like really white to almost an electric blue coloration. It has that hump, and I'll show you a profile photograph here in just a moment that really makes it kind of stand out. Other species in this family don't necessarily have that, but they're relatively small, so they're only maybe a little bit over an inch to inch and a half. So we're talking 2.5 to 4 centimeters in standard length, so not including the length of the tail itself. And we called it Demogorgon. So here in the U.S., I don't know how popular Stranger Things or Dungeons and Dragons is in Europe. So Dungeons and Dragons was a real popular, still is popular, basically kind of fantasy kind of board game. Not really board game, but a game in the 80s and 90s. So I'm an 80s kid that had a creature called the Demogorgon, right? This underworld demon. And so we wanted to, kind of wanted a name that would stand out a bit to draw attention, to these critters and cave life in general that really are out of sight now to people's mind. People don't know about them, even here, in these karst-rich areas like in northern Alabama. And so in more recent times, right, the Netflix series Stranger Things has a creature called the Demogorgon, kind of again based on this. So it's kind of a play on for both of those. Of course, ichthys means fish, and then the specific epithet arcanus, Demogorgonichthys arcanus, is in reference to it being hidden for so long. So the fact that we've been, I say we again, the biologists in general, have been visiting the cave for going on almost 40 years, right? And no one really noticed, right, or paid attention to this fish that I'm sure many others have laid eyes on before, but we just didn't pay attention to, didn't notice. So here's what the fish looks like in profile. You can kind of see that hump. We call that nuchal hump, kind of a fatty hump behind the head in general, and that kind of sloped skull. You can see the individual kind of scales there in the body, but again, it kind of lacks pigment. In contrast, kind of go back and forth. This is what the southern cavefish, the other fish that occurs. This is actually a picture from a fish at Bobcat Cave. So if I kind of go back and forth, you can now you kind of see those differences. They do look quite different, but if you're looking, looking from a pool, you're standing a few feet above it, right, shining your light. It's can be kind of subtle. You're not really paying attention, to those differences, and I think that's part of the reason why it remained, undetected for so long. And this is some more information, yeah, in terms of their overall. We have very limited information about its ecology and its life history. At this stage, we're hoping to study it more. As I mentioned, we have roughly about a 1-to-1 ratio, and that's what really is kind of unusual. These are fish that are living. The two species are living side by side with each other. If we're not having a situation where it's like 90-95% one species and very very few or very rarely you kind of the other species. They appear to be about equal ratios, which again is something that we typically don't see. Some other aspects of your if you're a fish nerd and into their aspects of the morphology, some of the differences between the southern cavefish and Demogorgonichthys there, the center, the presence versus absence of like the papillae, certain sensory structures associated with the lateral line system, there on the head, head region, that hump we mentioned. We talked, both black eyes. Typhlichthys still has those melanophores. So if I go back to that one picture, it almost looks dusky. You can actually see the myomeres, right? Those are actually the pigment cells that are bordering the myomeres that are being expressed. So we brought this fish up to the surface briefly. Within about 5 minutes, it already starts to express that melanin and kind of gets that dusky appearance. Demogorgon, yeah, the demon cavefish doesn't do that. So this is a figure that we put together right for the paper, but kind of showing, these are the three species that occur in northern Alabama now. Southern cavefish, unknown cavefish at the time. This is the demon cavefish now, kind of between, and then that duck billed, right, the Alabama cavefish there on the bottom. And in a way, it almost looks like the demon cavefish is intermediate, kind of in the morphology between those two other species. Here's another kind of comparative shot of their heads, just to kind of show how they differ. Again, the demon cavefish there, in the middle. So you're seeing maybe this trend, in this group of fishes that they're kind of get a progression. Perhaps this is indicative of how long they've been underground, isolated underground, but elongation of the jaw itself and changes to that cranial morphology over time. And so, one inference we might be able to make is, perhaps the Alabama cavefish there on the right has been underground longer than the demon cavefish there in the middle versus the southern cavefish there on the left. So here's a example of some of the micro CT scans, again, kind of also illustrating those differences. So a lot of the bones are still present. They're just slightly different in their orientation and structure in general. Again, the top is the southern cavefish, middle demon cavefish. The bottom is that Alabama cavefish. From a genetic perspective, and this is where really things were surprising to me. When we first started getting some of the morphological data in, I honestly thought it would be a species we probably would place in that genus Bioplatyrhinus with Alabama cavefish, just with the trend towards the elongation of the head, some of the other features, the unbranched fin rays, for example. When we started to get the genetic data back, including we amplified entire mitochondrial genomes, comparing that with some of the other amblyopsids, and finding essentially 11 to 12% or more sequence divergence across the entire mitochondrial genome really was surprising. So this is what this is kind of showing here. There's some other unique aspects of the mitogenome that were different, and then we also looked at the I gene rhodopsin. So in some of the other amblyopsids, this is the gene that's associated in vertebrates with your light dark vision. So you have this gene in your eyes as well. It gets expressed. It's been knocked out in a few cavefish species, and including things like the Ozark cavefish. In the demon cavefish, it has a really unique signature of loss of function and functionality. I'll talk about a little bit. So here's the essentially the phylogenetic tree. And this is a time calibrated tree. So branch lengths correspond to amount of divergence. And there in red is where the demon cavefish kind of came out as. It's essentially is a polytomy. So essentially kind of a branching over the same time period as southern cavefish, so the genus Typhlichthys, and then that Speleological is the Alabama cavefish around maybe 11 million years ago. So our confidence intervals anywhere from 10 to 13 million years. So this isn't something that, has been isolated for, the last few 100,000 years or the last couple million years, maybe since the last ice age. This looks to be a lineage, right, that's been on its own trajectory for a really, really long period of time, which is really, really fascinating, to me. So here's kind of summary of that rhodopsin I gene. So at Bobcat Cave, the southern cavefish, it technically has a functional rhodopsin. Whether it actually utilized is something we don't necessarily know real well. But in the demon cavefish there, it's actually missing about a third of its coding region. Has a 284 base pair deletion that also causes a frame shift that leads to additional, basically, loss of function mutations, premature stop codons that are expressed. So basically wiping it out entirely. So it's not being used. And again, this is a trend that we do see, and we're starting to understand more in eye or pigment-related genes in a lot of cave organisms, species that maybe have been isolated for longer periods of time. You don't necessarily need eyes anymore, right? So why maintain the genes that are associated with that? And so this is evidence that we're seeing maybe relaxed natural selection on these features. And another great example of convergent evolution. So we've got, they're coming from, a similar genetic background in that they're at least in the same family, but they're kind of converging on a similar solution morphologically, right? And that might be, again, partly why the demon cavefish remain undiscovered for so long, because it does morphologically look still very, very similar to southern cavefish. And white still is kind of surprising, and that we've we found this new genus, not just a new species, is that at least in the eastern North America, our freshwater fish fauna is fairly well described. We're still finding new species. Often it's cryptic species, or it's something's been there's been some genetic work that shows, oh, this particular population that's in this watershed here, or this river drainage, right, is genetically distinct and it's on the evolutionary trajectory. We see those types of studies happening quite a bit. You really kind of have to go to Southeast Asia, Borneo, other areas like that to find, really new, families or genre of fishes. And again, something that we were not expecting, going into, even when we made the initial discovery and initially kind of talking about this being a new unknown species and describing it. As far as we're aware, we do know, right? It's the first species or new genus of amblyopsid cavefishes since 1974, and that was with the description of the Alabama cavefish. And talking with a colleague, with Mike in particular, this might be the first, we think, genus in the last 50 years in the eastern U.S., which is pretty amazing. And this is like these are cavefishes here in the eastern U.S. Anyway, this family that had been known about. They were the first species. The northern cavefish was described from Mammoth Cave in 1840. So we're talking, almost 200 years now. But in terms of being kind of a model system for cave biology, really since Eigenmann's work, Carl Eigenmann, who was an ichthyologist at Indiana University, starting in 1898, 1900, was the first to kind of use this system as kind of a comparative morphological model for cave adaptation. So it's relatively well known compared to other vertebrate cave organisms. So I've already kind of hinted at this, how did it hide for so long? Partly it's related to the cave that it's in. It's a flashy system. We can't necessarily access a lot of that cave throughout much of the year. Even if you can get into the pools, it has a pretty thick mud and you can stir that up quite quickly. That leads to reduced visibility. And we try to stay out of it in general. And so because of the shrimp are there to minimize that disturbance. And so, these factors, and as well as we've known about southern cavefish being there, the previous collections that are in the Museum of Sessions, all are southern cavefish. No one had collected one previously. No one was assuming that, hey, there's something new in this cave system. A little bit about its conservation. So as we're aware, right now, it's still just known from Bobcat Cave. We have visited other nearby cave systems since the discovery to see if maybe we overlooked the species in other caves. And as far as we can tell right now, we haven't yet. That doesn't mean that that's not the case, but it's just known from the single cave system. It has a recharge basin of maybe only half a kilometer squared, pretty small area, but I suspect the fish is probably accessing a deeper aquifer. Exactly how large is its habitat extent, we have no idea at this stage. We don't know anything about its ecology, its life history, how long do they live, how many offspring do they produce, how often do they reproduce. All those types of questions are still unknown at this stage. In terms of threats, some of the bigger issues is, this is an area that was involved of the military installation that had previous, ordnance and munitions, testing. And so in the past, there has been elevated lead, cadmium, some other heavy metals in the groundwater there. And one impact that's had on the population of this cavefish and other cave life there is really kind of an unknown. It is strongly linked to, the surface. It rapidly responds hydrologically. So there's a lot of development on the western edge of the arsenal, not on the arsenal itself, but just off that residentially over the last 2 decades. Some of that probably contributes to the recharge of the cave. And so these are all things that potentially could impact the species in general. So just kind of quickly summarize them back out here. Again, another example of cryptic diversity that we see in cave biology. A little bit different story on this one. It's not simply it's like something that we're splitting up that was well known. This is entirely something that was was hidden for a while. But I think a bigger point is just because we have studied certain caves, right, and been visited a lot of them, we might refer to them as being well studied. That does not mean we still know everything about these systems, and so we see this detection bias in general. And having long monitoring doesn't necessarily guarantee that you know everything about a particular site or about a particular cave system in general. It also, kind of highlights, here in our area, probably should maybe include like a satellite imagery and just kind of the land use change that's occurred over the last 2 decades here. Huntsville's a rapidly growing area. Um, that's one of the fastest growing cities in the United States. People are buying up land, subdivisions are going in, clearing land in general, and so having larger tracts of at least relatively protected land is fairly rare. And the military installation here at Redstone Arsenal actually does offer that in the buffer zones, around some of their other testing areas and other sites, sensitive sites. And so, it kind of highlights these military installations can be important for, sheltering, undescribed biodiversity, in general. So key takeaways, again, this new discovery of a new genus and species. Really, despite the fact that we as biologists have been monitoring it for 30 plus years, almost going on 40 years now. It was just assumed to be the southern cavefish, which is a more widespread species here in the eastern U.S. Both of these species, the Typhlichthys, southern cavefish, and demon cavefish, they've converged on a similar kind of morphological, I guess, solution to living in complete darkness. But it is an endangered species. Doesn't have any sort of state or federal protection yet. We'll see if that materializes, down the road. Kind of next steps is for us to try to better understand its distribution. So using things like environmental DNA, for example, so we can detect its presence at other sites, and then use that to guide more targeted survey efforts in other regions. Try to learn more about its ecology, and life history. And another point, I want to make and kind of end on is the importance of, in this case, is driven by biologists, but a lot of our understanding, particularly groundwater life and just cave life in general, first comes from cavers like yourselves. It's someone noticing, oh, I thought I saw a cavefish here. I think I saw a beetle, in this cave, and something looked a little different, interesting, reporting that. That's often how these discoveries first get made. Where my lab's is currently involved with several other new discoveries that we're going to be publishing on different parts of the eastern United States, and in almost all these cases, it involves cavers like yourselves who first are making those discoveries, and we're working with folks like you on getting access to these sites or helping with logistics and leading expeditions to try to collect specimens, try to collect data, right? So we can better understand, this life that is largely hidden right now to sight from most of the world. So with that, so a few other acknowledgments. Again, my co-authors again. This work is permitted through the U.S. Army as well as the state of Alabama. Funding sources: the U.S. National Science Foundation, USGS provided funding from previous trips, state of Alabama in general. The paper itself, there's a link here. It should be open access. Anyone should be able to see it if you're more interested and would like to read the paper. I'm also happy to answer any questions you all might have. Here's my contact information. Find me on Instagram, or Facebook. My wife and I have a company, Niemiller Nature, that's also involved with this as well. But with that, I'd be happy to answer, assuming we've got some time, any questions that you all might have. And thank you for listening.
Thank you so much, Matthew. This was great. Actually, I already have a question. I wanted to ask you, what was your personal reaction when the results confirmed that actually we are looking at a new genus?
I was really surprised. Like I said, going into it, wasn't suspecting or even thought it would be a new genus. We got the molecular data back that suggested that, hey, this thing looks to be quite old and isolated from both the southern cavefish and Alabama cavefish. That's when I was like, wow. And even then, it didn't really hit me until our colleague who's co-author too, Mike, made a point. It's like, well, I think this might be the first genus, newly discovered genus in the eastern U.S. in maybe like 40 or 50 years. That was like, oh wow, that's really crazy. It just was really unexpected. So this has been one of the highlights of my career. I mean, I've published well over 100 articles, and this has been a real fun one. And I think another point to make too is, we first discovered this fish in February 2025, and the description came out last month in July. So basically 16 months, we were able to go from discovery to actually formally describing it and have that peer reviewed and published, which is only possible because of the team that was involved, and including also the students. I definitely want to give them, give them their praise and credit. I mean, it was a team effort. All the labs that are involved, and I've got to have a long history with John's lab and Pam and Mike as well. So, and hopefully we can continue to work together on future discoveries.
Okay. And also, in terms of the cavers who are not biologists, who don't know what to look for underground, or they see something, an animal or a fish, and they have no clue. Uh, how do we recognize it? What should we do? What should we
Sure,
definitely not
great
do?
question. Right, great question. So, I'm assuming if you're like most of us who are caving in the U.S., we almost always take our cell phones with us underground, or we have some sort of camera right with us. Sometimes the first thing is just if you can get a halfway decent photograph of what you saw, and even if it's something common, that's useful information to report. Oh, it's a it's a cricket, a beetle that's actually from the surface. That's still useful information. If you see something that you think is, really, really unusual, finding, a local contact, who might be a cave biologist, there in your country, in general is kind of the first step is, a lot of cavers that I work with or that I know, some of the Facebook groups, they'll send me a message, right, or an Instagram message like, oh, I saw this salamander larva in a cave. Can you identify it for me? And that's sometimes how it starts, right? Or it's like, I think I saw a weird fish here, right? Or I saw a cave crayfish. And that's how a lot of our new records first, are reported. And, if you have an interest in cave biology, taking some notes just as in your caving trips, like, oh, I think I saw some bats, right? I saw four bats that I think were this species. If you can get a picture, even better, right? Or I think I saw, a cave beetle. And even if you didn't get a picture, reporting that is really, really useful. One of the things that I'm trying to work on here in the US is actually creating a data portal where cavers and others can actually fill out a form and report some information, upload photographs that can then be vetted. That we can try to identify that because there's not many cave biologists, operating in the U.S. There's not that many in Europe, either. But there's a whole lot of cavers that are going underground, and you guys are the first lines. Sometimes you're the only ones that can get to certain areas of the certain cave systems, right? Or maybe your relationships allow you to get access to a site that others can't get to. Um, that's, I think one of the best ways in that kind of developing this network and maybe a reporting system to try to acquire this data a bit more easily. iNaturalist, if you're familiar with that, is another venue that we sometimes that I try to pull some data from. It's you see that more and more commercial caves and more publicly accessible caves. We get a lot of reports there, not necessarily on private lands. One of the concerns that we have here in the U.S. is cave data is still quiet, or cave location data is still very, very sensitive. And often folks don't like that being shared by folks. It could be state agencies, it could be actually caving clubs or caving grottos and whatnot. And so we have to be sensitive and mindful of that. But yeah, I mean the more awareness we can have and more eyes looking, I think is really, really beneficial, so we can better understand these species. Um, one of the things that my lab's trying to do is I don't like to see species listed as being endangered or threatened. In some cases, it could be because we just don't have the data. We only know them from one site because no one's really been looking for them before. We need to, try to determine, well, is it truly rare, or is it just something we've been overlooking? And that's only possible through repeated visits.
And last question on my side. I was wondering if you have any data of how vulnerable is the Damon cavefish population? How many individuals are
Right,
they?
right. Mhm. Yeah, that's in terms of overall population size, we just have our census count data. Our last trip there a couple of weeks back, we did have our largest count of 27 fish. That doesn't mean that I'm sure there's
27.
many, many more, but yeah, we've not conducted a population or marker capture type study to estimate population size effectively yet. That's something that hopefully will transpire here in the next couple of years. We're still kind of in the exploratory phase of, hey, can we find it anywhere else nearby? And that's where, as I mentioned, maybe using something like environmental DNA as well as traditional visual surveys, and going back to some of these cave systems where we have reports of cavefish to confirm, is it just the southern cavefish there? Do we see both species there? And so on is going to be a priority here moving forward. So yeah, I guess stay tuned. Hopefully we'll have some more information, relatively soon.
Okay, cool. Now I finished. If anyone else has questions for Matthew here. Okay. So if you don't have questions anyway, you will be able to access the recording. I asked a bit on our communication groups because I was a bit impressed by a small presence, and it seems that everybody is out in it's August, so everybody is out in caving trips and expeditions. Oh, I can see a hand raised. Colleen, if you have a question. We can't hear you. Maybe something is wrong on the connection. Microphone.
So worst case, if you could type your question if you have a question for me. Happy to answer.
Also, yes, we can use the chat. So until we I can see that Colleen would want your context. Definitely, it's also here on the last slide that is presented. And the question is if you found salmonids in caves.
So yeah, here in the U.S., this is definitely the eastern U.S. The only situations where you find any salmonids are on trout farms, and so yeah, they're not usually in that case native to the cave system itself. It's fairly rare, but yeah, it does occur. We don't have any cave populations. There's a couple commercial caves here in the eastern U.S. One that's called the Lost Sea in eastern Tennessee that talks about having cave fish right in their cave system, and they have a boat ride to go out. They've stocked their cave with trout, and they feed them regularly. So that's not really the same thing, but yeah, unfortunately, no. Out west in the western U.S., there's a few records I think of some trout maybe kind of wandering up in the caves, but they're fairly visual predators. And in general, much like our centrarchids, so our basses and sunfishes, they're very visual, and so there's just not any cave adapted species for the most part for those groups.
Okay. Anyone else? Okay, then. So the recording will be available on our YouTube channel, and I want to thank again to Matthew, the director of the Cave Biology Lab at the University of Alabama in Huntsville, for this amazing presentation. And I also want to congratulate you for your hard work and discovery. And also, please tell your team that from the Balkan Speleological Union, we are very proud to have this information from you.
Oh, thank you. I really appreciate it, Emma.
Thank you very much. So have a nice evening. And I don't know if in every country you can see the solar eclipse, but I think you can still catch a few minutes out of it.
Yeah, I'm gonna try. I don't think where I'm at, we're gonna be able to see very much, but we'll try to take a quick look.
Okay. So thank you all, and bye-bye.
Take care.
Bye-bye.
Bye bye.
Bye.